Good day, students. We'll continue our class on the solutions to 2019 SSCE. Now we're on question 5. So what stage of cell division is illustrated in the diagram? So this diagram is a diagram of a stage in cell division. For example, we have the mitosis. Now, let's look at the stage or what phase of the mitosis we have for this diagram. A, prophase; B, metaphase; C, anaphase; then telophase. So let me just give you the major event that occurs in each of the stages, then we'll pick our answer. The prophase, the major event that occurs in the prophase is the chromosomes become visible as two chromatids. Each chromosome becomes visible as two chromatids. Number B is the metaphase. The metaphase, the chromosomes align at the equator, and you can see here that the chromosomes here, they align at the equator. So the answer is metaphase. However, the anaphase stage is the stage in which the chromosomes move to the opposite poles after they have been separated at the middle here, at the centrum here. They've been separated at the middle. The middle is called the centromere. The centromere joins each of the sister chromatids together in a chromosome. So the telophase is the phase in which the cell becomes constricted to form two daughter cells. So let's go to the next question, number 6. The part labeled 1 is the dash. So in this diagram, we need to analyze some major parts here. The first one is you have a radiation coming out of this structure. So the radiation coming out of the structure, if it's what is meant in this diagram, the answer is going to be the aster. The asters are smaller microtubules that radiate out of the centriole. So the centriole is the middle here. This is the centriole, and we know that the centriole is found only in the animal cell, and the function is that it helps to produce the microtubules or produce the spindle fibers that assist in the splitting of the sister chromatids during cell division. So these fibers here that are sent out they are produced by the centriole is called the spindle fibers. It's called the spindle fiber. So in this question, if the diagram points, labeling points at this structure here, the answer is going to be aster. So if it points at the central, the answer at the middle here is going to be the central. So let's look at the options. The part labeled 1 is the dash. Centromere cannot be the answer because the centromere is what joins the sister chromatids together in the chromosome. So the spindle fiber, no, these are the spindle fibers. The spindle fibers actually join the centriole from opposite pole together and also separates the sister chromatids from the chromosome during cell division. So the answer cannot be centromere. And the answer can also not be spindle fiber for now. The answer can be centriole, and the answer can never be chiasmata. Chiasmata is that region where the chromosome exchange genetic material. Okay, the homologous chromosomes exchange their genetic material. So the answer here, as I choose, is going to be central. Now let's go to the next question. Question 7. The rate of diffusion of molecules between two media will be higher if this is talking about what factors increase the rate of diffusion in biology. A, the difference in concentration of the two media is low. Yes, when there is difference in concentration, okay, let's have this. Let's have two containers, for example, okay, or let me just have a container so that we can get it better. We have the concentration of substances here, and we have the concentration of substance here. Here we see that is match. This is A, and this is B. There's a difference between these. Let's say we have 8 molecules here, we have 3 molecules here. The difference in their concentration, okay, is 8 minus 3 giving us 5. That is the difference in concentration, also called the concentration gradient. So if we also have another pot here, where the molecules are matched. Let's say, for example, 20. Okay, and here we have another molecule here. This is A, this is B. Okay, where we have 4A. So the gradient, that's the concentration gradient, is 20 minus 4, that is 16. So we're talking about where will the rate of diffusion be faster. So the answer is going to be whenever there is the difference between the concentration of A and B is higher than the rate of diffusion. It has a tendency of the molecules to move faster because of the much difference between A and B. So let's look at our options. Difference in concentration of the two media is negligible at all. If there is no difference in concentration between A and B, then the molecules are not going to move because the result or the end of the diffusion, when we define it, is for evenly distribution of the substances. So that means that the substances have been evenly distributed. We don't need to move them anymore. C, the difference in concentration of the two media is high. So the answer is C. Now let's go to question 8. Anaerobic respiration in the organism is illustrated in the diagram below. Produces carbon dioxide and dash. A, ethanol. B, water. C, oxygen. D, glucose. So the answer here is ethanol. So let me quickly run through the equation. And you know that this is yeast. Okay, this is yeast, and the yeast has the enzyme zymase that assists in the fermentation process. Fermentation process is the process where the glucose is broken down in the absence of oxygen to produce carbon dioxide, water, and a little amount of energy. So let me give you the equation here. We have glucose C6H12O6. There is no oxygen involved. However, there is yeast that has the enzyme zymase, and it's going to produce C2H5OH as the ethanol, and the carbon dioxide is produced while a little amount of energy is also produced. The equation is not balanced. If I put 2 here, I'm going to have 4 carbon here, and we have 6. Let me put 2 here also. Okay, 5 times 2 makes or gives us 10 hydrogen here. Okay, yes, we have a 10 hydrogen here, and we also have another hydrogen, that is 2. So the equation is balanced. So we meet in the next class. Goodbye.